Hi to all. Marchantia reproduction, vegetative and sexual reproduction. So in last class we are already discussing about the Marchantia vegetative characters and the structure of gametophyte. Um, so in this lesson we are discussing about the reproduction in Marchantia. So in Marchantia the reproduction takes place by two ways. One is vegetative, another one is the sexual reproduction. So the vegetative reproduction in Marchantia. Vegetative reproduction takes place by the following methods: by progressive death and decaying of the thallus. It is a common mode of vegetative reproduction in bryophytes. In like Rixia, the vegetative propagation of Marchantia quite common, which takes place by unlimited growth of branches and decay of the base. When the decay of the cell reaches dichotomy, the two lobes become spread from each other and each separate lobe form an independent plant. The second method is the adventitious branches. These branches are developed by the ventral side of the thallus. They are look like the reproductive buds. They detach from the parent plants and grow into the new plant. The third method is very important and common one markant, that is the vegetative propagation takes place by gemma. The most common method of vegetative propagation is specialized asexual multicellular bodies known as the gemma. They are born in large numbers in small receptacles called gemma cups present on the dorsal surface of the thallus in the midrib region. A gemma cup is about 2 mm in diameter and 3 mm in height, the margins of which are haline lobed spinous or entire. Many small stalked gemmae developed on the floor of the gemma cup. Each gemma is vertically attached to the base of the gemma cup by one celled stalk. The gemma are multicellular, biconvex, discoid bodies constricted at the middle. The two notches in the constriction possess a row of apical cells showing two growing points. So sequentially, the apical cells present in the two notches grow to develop two thalli of new independent plants opposite direction. Usually, gemma present on the male thallus produce male plants. Those on the female thallus form female plants. So this is the gemma cups. So the the TS of gemma cups are look like this. These are the gemma. Each gemma is look like this shape. These are the notches. From these notches, the germinating cells are developed. From each gemma, two plants are developed. This is the vegetative propagation in Marchantia. The next, sexual reproduction. The sexual reproduction is takes place by the two sex organs, that is antheridium and archegonium. Antheridium is the male reproductive organ and archegonium is the female reproductive organ. So in Marchantia, the production of sex organ is dependent on environmental conditions like day, length, humidity, excess of nitrogen substance, etc. The sex organs are born on special stalk receptacles called gametophores. So antheridium develops on the antheridiophore and archegonium is developed on the archegoniophore or carpocephalum. These are developed on separate plants so that the Marchantia is dioecious or heterothallic. Although the gametophores are erect branches, they are actually indirect extension of the prostate vegetative thallus. This is clearly evident from the rosy ventral nature of erect shoots with air chambers, air pores and rhizoids. So antheridiophore, this is the vertical branch arised from the thallus. Now you see the TS, the antheridiophore. The antheridiophore shows long stalk bearing an apex slightly convex. Disc is usually 8-lobed structure. These are present along with the upper dorsal media in the antheridia are born on the row. The antheridia develop acropetal in manner. The oldest ones are bearing center and the youngest ones are at the peripheral region. That means the oldest ones are in the center and the youngest ones in the peripheral region. So antheridia, these are Marchantia dioecious and heterothallic. Although gametophores are erect branches, the mature antheridium has a short stalk-like globular body encircled by sterile jacket which encloses a number of sperm mother cells. A single sperm mother cell forms two triangular androcytes, each of which develops a biciliated sperm or anthrazoid. The dehiscence of antheridium in Marchantia is similar to the Rixia. Now you see the archegoniophore. Archegoniophore is look like this, like a lobed structure. It bears the archegonia. So the archegoniophore or carpocephalum is composed of a stalk and a disc bear 9 rays instead of lobes. In the early stage, the archegonia develop on the upper side of the disc. About 12 to 14 archegonia are arranged in a single row, each ray of the disc. The stalk of the archegonium being elongated just after fertilization, the central sterile part of the disc starts enlarge enormously. As a result, in the marginal apical region of the disc containing archegonia are pushed down to the lower side of the disc. So that's why during maturation it looks like the ray-like structures. So the mature archegonia at the peripheral side and the younger archegonia at the central side due to the formation of lobes. So rays are long, short and green finger-like projections that give a mature female receptacle an umbrella-like appearance. The archegonia become inverted by the curvature of the disc. Single-layered plate of tissue overlaps on either side of row of archegonia to form pericaecium or involucre. This plate of tissue encloses all archegonia arranged in a single row. A mature archegonium is a flask-shaped structure. It bears two regions. One is the venter and another part is the neck. The venter contains two cells, venter canal cell and the egg cell. The neck is possessive, it contains the neck canal cells. So neck canal cells is elongated neck and the venter cell and egg cells are seen in the archegonium. So now the fertilization, the anthrozoids of the antheridiophore discs are splashed by rainwater on the archegoniophores. The rest of the procedure of fertilization is the same as a Dexia. The archegoniophore is further developed in size after fertilization, which eventually leads to the inversion, upside down position of archegonia. In this phase, the development of rays and pericaecium takes place. The fertilized egg enlarges in size and fills up the cavity of the venter. It invests itself immediately within thin cellulose walls, become oospore or zygote, which the mother cell of the sporophytic generation. So during fertilization, what happens means all these the neck canal cell and ventral cell completely disintegrated. The egg cell only present. At that time, the antherzoids moves through water current, and they reaches the archegonium and fuses with the egg cell and form the zygote. This is the sexual reproduction. From this stage, the sporophytic phase takes place. So these are in the life cycle of Marchantia, there are two phases. One is gametophytic phase, another one is the sporophytic phase. From the formation of zygote, the sporophytic stage starts. Thank you.